And now we discuss the another important group of higher fungi. These are Basidiomycetes. They actually make club-like bodies, so that's why they are also known as club-shaped spore-bearing fungi. These fungi are also important because they are known for lignin degrading, lignin degradation. Lignin is an important cementing material present in wood. Then actomycorrhizal fungi mostly belong to Basidiomycetes. Then important character because of which they are known as Basidiomycetes is the specialized structure known as basidia. Basidia are these club-shaped bodies on which the spores are produced, and mostly 4 spores are produced on each basidium. Then another important character in Basidiomycetes is the presence of 3 types of mycelia, that is primary mycelium, secondary mycelium, and tertiary mycelium. Primary mycelium is monokaryotic. It contains only 1 nucleus. And secondary mycelium are basically dikaryotic. They contain 2 nuclei, and the 2 nuclei are different from each other. And then tertiary mycelium contain the club-shaped bodies, or mushroom-like bodies, on which the basidiospores are produced. Another important character of Basidiomycetes is the presence of barrel-shaped septa in mycelium. These barrel-shaped septa have dual pores on each side. And another important character of Basidiomycetes is the presence of clamp connection when they divide when the cells divide. During each division, clamps are formed between the 2 cells. So if I conclude all this, so Basidiomycetes are the fungi which are having club-shaped bodies on which basidiospores are present, and these club-shaped bodies are known as basidia. Then these Basidiomycetes are present in 3 stages. Primary mycelium, secondary mycelium, and tertiary mycelium. They have barrel-shaped septa in their hyphae, and each septum have 2 pores, one from one side and one from other side of the barrel. They make clamp connection when they divide. During their division, clamps are made between 2 cells. So you can readily identify the Basidiomycetes on the basis of presence of clamp connections and barrel-shaped septa. So Basidiomycetes are important because of their role as a saprotrophic fungi. They grow very well on dead logs and manures such as cow dung, and they also cause kind of problems in turf grasses, such as they cause dry patch and fairy rings because of their underground growth. Then a number of Basidiomycetes are important parasites in especially in plants. Like Rhizoctonia is an amorphic form of Thanatophorus, and the Rhizoctonia is a very important pathogen in a huge group of plants, diverse kind of plants, and they cause losses. Then another important group of pathogens like that are known as rust. Rust causes problem in diverse plants, especially in cereals. They cause important losses, and rust is caused by Puccinia species belonging to Basidiomycetes. Then another group of pathogens is called smut. Smut disease is caused in plants. They are important in cereals. They belong to Basidiomycetes. Then Basidiomycetes are important. You cannot forget the role of mushrooms in food industry. They are rich source of proteins. Now we discuss the habitats of fungi in soil. The fungi are cosmopolitan. So most of the fungi are present in soil. They are present in all types of soil. As I have already discussed this point a little bit, that the hyphal growth best suit the soil environment because they start their growth from point source of nutrient, and then from there it grows to some other in search of some other nutrient source. So their hyphal growth can sustain their growth. I mean, in finding and help them in finding nutrients and fresh sources. And then the places with low nutrients and less moisture are also suitable for these for fungal growth, having more aeration. The fungal abundance and their diversity also change with climate and altitude. The areas where temperature rises contain different types of fungi as compared to the areas where there is low temperatures with high altitudes. Similarly, the harsh conditions promote the fungi to produce some kinds of survival structures. The survival structures are mostly spores. However, there are some fungi which produce dormant structures like sclerotia. These dormant structures help fungi to survive for a longer period of time in unfavorable conditions. Then, as I have already discussed a little bit, that fungi play important role in soil, and they are sometimes major biomass of microorganisms in the soil. The accurate detection of soil, accurate detection of fungi in soil is always a challenge because of complex nature of the soil medium. And then aggregates are different. The difference is in a very minute level, is at a very minute level. Some aggregates are of different shape, some others are of different shape. I mean, there are difference in shapes, then there's variability in water and nutrient availability. So the microhabitats are created at very short distances. So there are two types of methods through which we can detect the soil fungi. There are direct methods and then there are indirect methods. So when we say direct methods, it means we see or detect the fungi directly, for example, using confocal laser scan microscopy, then using digital imaging and analysis. There are a number of fluorochromes that are available for accurate staining and analysis. Separating these fluorochromes help in separating live and dead fungal hyphae. Then there are methods for hyphae separation, through which we separate hyphae from soil, and then there are methods for staining. For example, the hyphae can be stained by diazonium blue. The different types of different groups of fungi can be separated, including ascomycetes, Basidiomycetes, and Zygomycetes, based on their morphological characters. For example, Basidiomycetes have very prominent clamps on their mycelium, so they can be separated from other groups. Similarly, Zygomycetes are aseptate, so they can readily be identified and separated. So in this way, we can separate the different groups of fungi. Then if we are using a specific type of fungi that we're studying in an experiment, so we can detect that fungi in the environment by specially designing a fluorescent level antibodies for that fungi. So we can detect this fungi in the environment. So then there are indirect methods for studying fungi in soil. For example, there are different molecular techniques that in which some specific signature molecules are targeted. Most important molecule is the nucleic acid. Nucleic acid can be extracted from the soil, and then specific primers can be used for detection of fungi. Similarly, enzymes, we can work with enzymes also that are produced by fungi. Fungi can produce extracellular enzymes. These extracellular enzymes are produced in substrates, different substrates. The purpose of producing these extracellular enzymes is to digest the organic matter, to extract energy out of that. Then there are other molecules, for example, phospholipids. Ergosterol is a specific sterol that is solely produced in fungi, and this sterol is rapidly degraded upon death. So this character can be used to analyze the live fungal biomass in soil. So we can extract ergosterol from soil and analyze. That will be a sign of living fungal biomass. And then we can supplement soil with antibiotics or antifungal compounds. This depends on our experimental design, what we want to do actually. So if we want to remove fungi, then we can use antifungal compounds, or if we want to remove bacteria and only we need fungal biomass, then we can use antibiotics. Then we can use glucosamine contents as a signature molecule. Glucosamine is present in the fungal cell walls. It's a building block of chitin. So now, finally, I revise the role, in brief, the role of fungi in soil. The most important role is decomposition. It's an important decomposer in soil. It decomposes litters and wood logs. It's the most important decomposer of lignin. Lignin is the cementing material of woods. Mushrooms are Basidiomycetes that are known for lignin degradations. They have important role in carbon nitrogen cycles. They are able to produce large amounts of exoenzymes or extracellular enzymes. Then they play important role in symbiotic relations in plants. Mycorrhizal relationships are something which are very considerable in whenever we talk about plants. Then they make symbiotic relations with other organisms, like they make with algae or blue-green algae. Such relations are known as lichens. Then they have mycoparasitic relations with other fungi, for example, relationship of Trichoderma and Rhizoctonia. Such relations have been explored frequently to search the biological control agents. Then the higher fungi have some role by feeding on bacteria. For example, pleurotus is a mushroom that attack on colonies of bacteria. Pleurotus also feed on yeast. So it's a kind of predator role that it is playing. Then the most important pathogens of plants are belonging to fungi. The most important diseases of plants like root rot of plant, damping of young seedlings, and fruit rot and rust diseases, all these pathogens belong to fungi. So thank you very much